This Meet the Professors session, I discussed briefly the recent updates on CAR T-cell therapy in lymphoma. So to briefly to summarize, I've discussed some of the results from CAR T-cell therapy in aggressive B-cell non-Hodgkin lymphoma, mantle cell lymphoma, as well as indolent B-cell lymphomas. In terms of aggressive B-cell non-Hodgkin lymphoma, there are 3 clinical trials that have been completed, multi-center pivotal trials. 2 of the products have already been approved, Axicabtagene and Ciloleucel, resulting from the ZUMA-1 study and the JULIET study. The TRANSCEND study evaluated Ciloleucel product, and this also showed, this was also completed and recently published, and the product is expected to be approved later this year. So what these studies show uniformly is that in patients with relapsed or refractory large B-cell lymphoma, including DLBCL, primary mediastinal B-cell lymphoma, transformed follicular lymphoma, this therapy can be highly effective targeting CD19, with response rates in the 50 to 80% range and a complete response rate in the 40 to 60% range across these 3 products. What's also very interesting to note on these 3 trials is that a significant proportion of these patients seem to have long-term durability of those responses, approximately 40% or so. And we can see a plateau in the PFS curve at around the 6-month time point in these patients with very few relapses beyond that. In the ZUMA-1 study that has the longest follow-up to date at a median follow-up of 27 months, 39% of the patients remain progression-free, and almost 50% of the patients are still alive. In the case of the JULIET study, at a median follow-up of 14 months, 34% of the patients remain progression-free, and on the TRANSCEND study, 44% of the patients remain progression-free at a median follow-up of 12 months. In terms of safety, the most common toxicities that are noticed on these patients acutely are cytokine release syndrome and neurological toxicities, and these are mostly grade 1 and grade 2 across these 3 trials, with a slightly higher incidence of grade 3 toxicities on the ZUMA-1 study testing Axicabtagene related to the other 2 trials testing Ciloleucel and Ciloleucel that have a slightly different CAR design. But these toxicities are largely reversible within the first 2 weeks after the infusion. So I think overall these data suggest that anti-CD19 CAR T-cell therapy represents a major advance for the management of these patients with relapsed or refractory large B-cell lymphoma after at least 2 lines of systemic therapy. So these therapies are now being evaluated at earlier lines of therapy. There are 3 randomized trials ongoing in the second-line setting, testing CAR T-cell therapy head-to-head with stem cell transplant. In addition, CAR T-cell therapy in the frontline setting have also been initiated in patients with high-risk diffuse large B-cell lymphoma, as well as high-grade B-cell lymphoma, including double-hit and triple-hit lymphomas. In the case of mantle cell lymphoma, there's 1 pivotal trial that has been completed with ZUMA-2 trial testing KTE-X19, which is also now called as Axicabtagene or Ciloleucel. This has the same CD19 CAR design as Axicabtagene product, but with 1 minor difference in the manufacturing step, where the CD19 tumor cells in the apheresis product are removed prior to CAR T generation. So this trial was a multi-center trial testing 74 patients and showed a very high overall response rate as well as complete response rate at 93% and 67%. And at a median follow-up of 12 months, 61% of the patients remained progression-free. And on this trial, patients had to fail BTK inhibitor therapy as well as anti-CD20 and alkylating agent or doxorubicin-containing regimen. So this therapy also suggested that this trial also suggested anti-CD19 CAR T-cell therapy can be highly effective in relapsed or refractory mantle cell lymphoma, and this led to the FDA approval of Axicabtagene or Ciloleucel in July this year in the US for relapsed refractory mantle cell lymphoma. In another pivotal trial, the ZUMA-5 study, which the interim analysis from this study was presented at ASCO this year by Karen Jacobsen. This was also a large multi-center trial with testing 140 patients at the time of the interim analysis, with a median follow-up of almost 15 months. There were a total of 80 patients with relapsed refractory follicular lymphoma after at least 2 lines of therapy who were enrolled on the trial, and there were 16 patients with relapsed refractory marginal zone lymphoma who were enrolled and treated and were evaluable for efficacy at the time of the interim analysis at ASCO. So this trial showed also that the response rates are very high in relapsed refractory indolent lymphoma with anti-CD19 CAR T-cell therapy. The overall response rate was 93% and the complete response rate was 80%. And at a median follow-up of 15 months, 68% of the patients were remained progression-free at the time of the interim analysis. And this trial also showed that the safety profile for Axicabtagene appears to be better than what we observed in relapsed refractory large B-cell lymphoma with the same product, suggesting that maybe the tumor biology or the tumor microenvironment could impact the safety profile in terms of CRS and neurological toxicity in these patients. So the primary analysis results from the ZUMA-5 study are expected to be present later this year at the ASH meeting and are eagerly awaited. Another publication that was recently reported in the Journal of Clinical Oncology in about a month ago, targeting CAR T-cell therapy targeting CD30 in patients with relapsed or refractory Hodgkin lymphoma. So this CD30 CAR T product was tested in 2 parallel phase 1/2 trials conducted at 2 different centers, Baylor College of Medicine and University of North Carolina. They treated a total of 41 patients, but gave different conditioning regimens among those 41 patients. Patients received either bendamustine alone or bendamustine plus fludarabine or cyclophosphamide plus fludarabine conditioning prior to CAR T-cell infusion. So in this dose escalation study treating 41 patients, the therapy was found to be quite safe. Patients primarily had grade 1 CRS. There was no neurological toxicity noted. One unique side effect noted with CD30 CAR T-cell therapy was skin rashes, which were observed in about half the patients, but most of these were grade 1 and grade 2 as well. In terms of efficacy, the overall response rate was quite high at 62%, and the complete response rate was 51%. And it appeared that in subgroup analysis, patients who received the bendamustine fludarabine conditioning appeared to have much better complete response rate as well as durability of responses compared to the other 2 conditioning regimens. So this led to a pivotal phase 2 trial that has been initiated with bendamustine fludarabine conditioning followed by CD30 CAR T-cell therapy. So that's ongoing at multiple centers now.